So what does that mean for cell line development? First of all, it means there can be quite some clonal variation in terms of afucosylation. That's what you can see here on the left-hand side in a plot. I think it's from Rose Glycard. You see, I think this is almost 250 or 300 different clones with, as you can see, different degrees of afucosylation. So yeah, quite a large span between starting from something like maybe 30 up to 60% afucosylation. And as a kind of binote here, you see how nicely that correlates with the affinity towards the Fc gamma receptor in an HPLC setting. So quite some clonal variation there. And on the right-hand side, you see in an approach from Genentech, where they knocked out the Fc gene, the FX gene, that different kind of clones respond differently to Foucault's supplementation in the media. So the general trend is kind of the same, but if you look at the red bars here, you can see they respond kind of differently. So also in terms of media compositions, clones react differently. So it's certainly in both aspects desirable to have screening technology in place that allows you to see the trends early on and quickly, so you can select, for example, clones that have the desired afucosylation profile. So let's look at the assays that we have developed at PAIA for this purpose. And I want to start with one slide that I guess, several people in the audience have already seen, so sorry for that. So this is the principle of our PAIA PA201 assay. As I mentioned briefly in the introduction, it's a bead-based assay, and in this glycan assay, we use a sandwich format. So we have a capture bead that captures the IgG, and then you have the glycan sitting on the Fc part of the antibody, and then we use fluorescence labeled lectins to bind and detect these glycans. This assay involves a sample prep step with a 5-minute heating, which helps us to expose the glycans on the IgG. The typical IgG sample concentrations are sometimes, well, somewhere between 50 and 200 micrograms per mil. The assay time for the whole plate is something like 90 minutes, which means that if you run afucosylation only screening and you run each sample in triplicates, you're able to run 96 samples in such a setting. And here on the bottom is depicted the workflow. This is a well. This is the labeled lectin that binds to fucose. You add the sample. As I'll explain later, that is a diluted supernatant. It's mixed on an orbital shaker. Afterwards, the beads are settling to the bottom, and then with fluorescence bottom reading, you determine the amount of lectin that has bound to the fucose on the IgG. So the lectins, as I said, bind to the specific sugar residues on the glycans, and we get binding rates for the different lectins. So this is kind of a setup that we would use in other glycan assays, where we have different kind of lectins for the different sugars: sialic acid, galactose, mannose, and fucose. So fucose is going to be the one that we would use for afucosylation screening process, obviously. And if we look at how specific this lectin is, one of course likes to enzymatic treatment of antibodies removes all the lectin binding, and we've done this here on this slide with two different kind of enzymes. First is PNG-ASF here on the left-hand side, and you see that all the lectins, all the binding of the lectins is removed by that. So that's PNG-ASF, and on the right-hand side it's endo-S2 enzyme, which leaves a little bit of glycan on the antibody. So you have these glucosnax here, and the core fucose is also still on the protein, and you see that still the core fucose binding lectin does not bind anymore, or just a very little amount. So it seems that this core fucose binding lectin really needs also some of the N-glycan core structure for binding. So that's usually 3 more mannose sugars which are located somewhere here. So that's the one experiment that I wanted to show you. And the other thing is the dynamic range of the assay. And for this purpose, we have generated calibration curves. And here I want to show you 2 examples where we mixed fucosylated and afucosylated IgGs. On the left-hand side, it's really an afucosylated IgG mixed with a fucosylated, and on the right-hand side, it's a fucosylated, kind of very normal IgG with almost 100% fucosylation, and then a variant of the same IgG with no glycans at all. And in both scenarios, you see quite nice calibration curves with an almost linear range of like 100% afucosylation to maybe 30, 40, whatever. And then there's a tendency that this curve kind of flattens out here towards 100% fucosylation. So for this whole range here, that's going to be the assay is quite nicely able to determine the different degrees of fucosylation. But you will have a hard time to differentiate between 90% and 95% fucosylation. Okay, so how do we apply these assays in a CLD screening process? So I want to show you an example of how we approach this. So first of all, what are the goals for afucosylation screenings in CLD? First of all, of course, we want to be able to perform a fast screening of potentially hundreds of clones. Those samples should be non-purified, so diluted supernatants, and we would like to be able to rank the clones with regards to the fucosylation levels and then do selection based on that. And of course, what would be really desirable, and that's what we can do with our assays, is that the screening can be done very close to the process in the CLD group where the samples are generated. So what do we need in terms of equipment for this? Orbital shaker, the faster the better, because that means the faster the assay is done. Then for the sample prep, we can either use something like a thermocycler, but you can also use an orbital shaker with a heating function, which is reliable. And then the last part, of course, is a reader system. Either that can be a fluorescence imager from Synentech, or also fluorescence plate reader can be used, like from Tek-Matic, from Molecular Devices. This is an instrument from BMG, exactly, yeah. So different kinds of equipment can be used to run the assays and to read out the plates. So